This PowerPoint will cover the microscopic anatomy and physiology of smooth muscle and cardiac muscle. Smooth muscle cells are called myocytes. They do not have any sarcomeres. They're not striated because their myofilaments do not align linearly. They have only one connective tissue surrounding them, that's endomysium. They are under involuntary control, meaning they are managed by the autonomic nervous system. They have a cylindrical shape that is narrowed and tapered at each end. If you look at the diagram, you can see what I'm talking about. They are uninucleated, meaning they only have one nucleus per cell because they're pretty small. And they are located primarily in the walls of hollow organs and blood vessels. There are two types of smooth muscle, unitary, also known as visceral, and multi-unit. If you look on the diagram on the right, you'll see the examples of the two. The unitary or visceral type is the cells you see are packed tightly together and they are arranged in sheets. Whereas the multi-unit, which is underneath it, you'll see that these cells are more individual and they have the ability to contract individually. Both types of muscle are innervated by the autonomic nervous system via cholinergic and adrenergic neurons. Both types lack a structured neuromuscular junction as with skeletal muscle. The terminal ends of the innervating neurons form bulbous swellings called varicosities that are analogous to the axon terminals of motor neurons. If you look at the diagram on the very bottom, you'll see an example of the swellings of the varicosities. I want to draw your attention back up to the unitary, aka visceral one, where you see a neuron going across a whole bunch of the cells and just dumping the neurotransmitters onto the top layer. That's basically how unitary muscle is stimulated, is just the top layer is stimulated. Whereas you look at the multi-unit, they are individually stimulated by their neurons. So some of the differences between multi-unit and visceral smooth muscle is that with multi-unit, each muscle cell is supplied by its very own nerve ending. These multi-unit ones are used for discrete, fine motor and localized motor control, such as the cilia of your eyes or the arachnoid pili of the hair follicles. The varicosities form what's called contact junctions, and they come in close proximity to the individual smooth muscle cell. Unitary muscle is the varicosities, as I showed in the previous diagram, will spread across the top sheet, and they form diffuse junctions. And because the smooth muscle cells of unitary muscle are connected via gap junctions, so if the top layer depolarizes, all the rest of the layers will depolarize via the ions being able to go through the gap junctions. And this type of muscle is used for more gross motor control of smooth muscle. Multi-unit smooth muscle cells are individual cells. They contract independently, but they can be recruited. A good example is pupil dilation or constriction. If you were exposed to a small amount of light, your pupil would be a certain size of constriction. But as the light increased in intensity, you could recruit more smooth muscle cells that would constrict it even further. The locations of multi-unit smooth muscle are the erector pili, that is attached to the hair follicle that causes your hair to stand on end when you have goosebumps. The iris and ciliary muscles in the eye that affect dilation and constriction of the pupil, and in blood vessels. In fact, blood vessels have both unitary and multi-unit smooth muscle. Unitary smooth muscle is also known as visceral muscle or syncytial muscle because it contracts as one unit. It is arranged in sheets, often opposing directions, as the example shows on the right of the intestines. We have longitudinal arrangement and circular arrangement. They go in opposite directions. The smooth muscle cells will contract as a unit, stimulating hundreds to 1,000 muscle cells at once that can contract together as one unit. The contraction of unitary smooth muscle is slow, synchronized, and extremely powerful. The force that's transmitted through one cell will be transmitted through all the rest of the cells during a contraction. Smooth muscle location. Well, they're located in the walls of all hollow organs except the heart. You'll find them in the respiratory tract, the digestive tract, the urinary tract, reproductive tract, and blood vessels, as I mentioned before, that also have multi-unit smooth muscle. The anatomy of smooth muscle. Very similar to skeletal muscle, but it has some differences. And I will be asking questions comparing the difference between smooth muscle, cardiac muscle, and skeletal muscle, and so will the board. So you'll need to know the similarities and the differences. I did post a short PowerPoint of 3 slides that listed a chart of the differences and the similarities between these 3 types of muscle. Smooth muscle has a sarcolemma, and they have a structure called caveolae, which is like a remnant of a T-tubule. They have a sarcoplasmic reticulum, myofilaments, just like skeletal muscle, actin and myosin. It has tropomyosin, but it does not have any troponin. And it has intermediate filaments, which is unique to smooth muscle, and another structure called Dens bodies, which is also unique to smooth muscle. The caveolae and sarcoplasmic reticulum of smooth muscle. The caveolae structures are pouch-like enfoldings of the sarcolemma. They have a large number of voltage-gated calcium channels within their depression. They could be a rudimentary T-tubule because they have a very similar arrangement as the T-tubules do in skeletal muscle, in that they are surrounded by the sarcoplasmic reticulum. The sarcoplasmic reticulum in smooth muscle is not quite as developed as it is in skeletal muscle, but it does store and release some calcium as the same function in skeletal muscle. However, there's no T-tubules running through the sarcoplasmic reticulum in smooth muscle. The myofilament arrangement of actin and myosin is the most unique feature of smooth muscle. They are arranged diagonally. That's one reason why it's not striated. This diagonal arrangement facilitates a very unique type of, and very strong type of contraction of smooth muscle. Actin and myosin of smooth muscle. The actin is a helical structure, just the same as it is in skeletal muscle, and it is stabilized by the tropomyosin, and its myosin binding sites are covered by the tropomyosin. However, it doesn't have any troponin that locks that tropomyosin in place. The myosin is shorter than in skeletal muscle and a little bit thicker. The heads along the entire length are oriented in different directions on each slide. I want to draw your attention to the diagram so you can get the image of that. They are in different directions. This causes a side polar cross bridge arrangement to the actin filaments. It'll pull the actin in different and opposite directions across the myosin, allowing for a very powerful contraction. The actin myofilament is attached to intracellular attachment proteins called Dens bodies within the smooth muscle. Smooth muscle doesn't have a sarcomere, so these Den bodies kind of act as the Z-line of a sarcomere. You look at the diagram, you'll see at each Dens body there's actin myofilaments attached to it, and the myosin is in the center of the arrangement between the Dens bodies. A little bit more about Dens bodies. They are an anchor for the actin myofilaments. They are analogous to the Z-line. Some are dispersed inside the cell, throughout the cell. Some are attached to the sarcolemma, and some actually bind to adjacent cells via intracellular protein bridges made out of connective tissue. and all are attached to each other through another type of protein, an intermediate filament. These intermediate filaments that are between the dense bodies form an intracellular protein bridge between the dense bodies. They are not contractile and they are arranged like lattice. You see the kind of crisscross arrangements. And they resist tension when there is a contraction of the smooth muscle cell. The intermediate filament dense body network is a term to describe dense bodies being held together by these intermediate filaments. If you look at this diagram, you'll see where it shows the connective tissue also attaching the dense bodies from one cell to another. And that is a key feature in allowing the smooth muscle to contract as one unit. This intermediate filament dense body network forms a strong cable-like intracellular cytoskeleton that causes the force of the contraction to pull through the entire muscle cell, causing the muscle cell to twist as it's shortened, making it look like a little scrunched up worm. The arrangement of this intermediate filaments and dense bodies transmits a pulling force to surrounding connective tissue. That surrounding connective tissue will pull on nearby cells, leading to a synchronous contraction or a syncytial contraction. And that is where all of the individual muscle cells contract together as one. Smooth muscle cell responds to stimulation from a lot of sources, from electrical stimulation, hormone stimulation, neurotransmitters, stretch, and even spontaneous stimulation. The same mechanism of contraction is the same for all types of stimuli, meaning no matter what has stimulated the smooth muscle, the physiology of the events of the contraction is the same. Smooth muscle contraction is very similar to skeletal muscle contraction in that actin and myosin slide across each other to facilitate a contraction. Also, the increase of intracellular calcium triggers the contraction process, and ATP energizes the sliding of this process. Mechanism of smooth muscle contraction. Extracellular calcium is the key to a smooth muscle contraction. Most of the calcium that's required to facilitate a smooth muscle contraction comes from extracellular fluid. Contraction of smooth muscle is dependent on extracellular fluid calcium concentration. There's no contraction if extracellular fluid calcium concentration falls below 1/3 to 1/10 normal levels. The mechanism of a contraction of smooth muscle, no matter what the stimulus is that voltage-gated calcium channels on the sarcolemma will open, allowing calcium to enter the cell from the extracellular fluid. If you recall, the caveolae have a high number of voltage-gated calcium channels, and that the sarcoplasmic reticulum is very close to the caveolae. Once the voltage-gated calcium channels open in those caveolae, it will also stimulate the calcium channels to open on the sarcoplasmic reticulum, dumping in a bunch of calcium into the intracellular cytoplasm of the smooth muscle cell. Calcium will immediately bind to calmodulin, and this activated calmodulin and calcium combination will activate a myosin light chain kinase, a specific type of enzyme. This activated myosin light chain kinase enzyme is the catalyst that will transfer a phosphate to the myosin, which will activate the myosin head. The activated myosin will now form cross bridges with the actin, and the contraction will begin.